Okay, so it's after 12, and we have 23 people on here. Thank you all for coming. I'm Eva Germain Shima Takahara, the president of this club for one more week. Our year runs from July 1st to June 30th, and Tim Novotny will be taking over after me. We are club number 334 in District 5110, and we were chartered in 1922. We have 115 members. Is that correct, Tom, or 113? Which is it?
We're down to about 101.
We had 115 before COVID. So we do have some visitors here. We have Sylvia Whitlock, the first female Rotary president in the world, joining us from California. And we also have Al Solomon. You have your wife. Can you introduce her?
Yes, Jean Adamson, who in a former life was a marine biologist. And of course, we're old friends of Trish's, so we're both interested in her show.
Great, welcome, Jean. Okay. And Trish Mace is on, and I will introduce her later. We will just tell you a few things about what we're doing here. Bill Bryle, previous governor, is co-host with me. This will be taped and put on YouTube, just so you all know. So to get started, let me introduce Trish Mace. Do we have any other visitors? No other visitors? Okay. In that case, let me introduce Trish. Trish is the director of the University of Oregon's Charleston Marine Life Center. That's an aquarium museum that highlights the diversity of marine life in Oregon. From 2011 to 2016, Trish was the manager of the Ocean Education and Partnerships at the Smithsonian's National Museum of Natural History in Washington, D.C. Prior to that, she managed the National Science Foundation, funded graduates in K-12 education at the University of Oregon's Oregon Institute of Marine Biology. She served on the Federal Interagency Working Group on Ocean Education, on the Centers for Ocean Sciences Education Excellence Council, and is a longtime member of the National Marine Educators Association. She's worked with a number of schools and nonprofit organizations on various ocean education projects, both domestically and internationally. Trish's interests span the fields of science, communication, and education in both informal and formal settings with a focus on ocean and climate science. Wow, Trish, that's a lot. I'm gonna introduce you. Can you come on right now?
So I just unmuted, and I just want somebody give me a thumbs up if you can hear me. All right, good. Okay, so I have a presentation which I will share in just a second. Actually, let me pull that up. I'm going to try sharing my screen. Oops. Eva, it says the host has disabled my ability to share my screen.
I put you on screen sharing. I see that right here. I wonder why not. Why not? Why is that not going on?
But I am seeing, I think, Tim's screen.
Let's see. I wonder why. I wonder why. New share. No.
I better stop playing games then.
Hey. I wonder why you're not coming in here.
This is his fault.
Let's go back here.
Okay.
Okay. Let me see. So, Bill, your co-host, can you look at that too? See why we're not getting her in here? I wonder why we're not getting her in here.
Try it now.
I got it. I got it too. I think she's on now. I got it. Share screen. Are you there?
Yeah, it's enabled. Okay.
There we go. Okay.
Oh, it says host disabled attendee.
No. Interesting. Let me go here again. Allow participants to share screen. Okay.
I just allowed it again.
I did too.
Okay, maybe it was toggling between. I'll try it again.
I see it.
There, it's there. Okay.
Good. Okay. Good. Good.
So is everybody seeing my screen now?
Yes.
Okay.
Yes.
Yes.
Okay. All right. Okay, so as Eva said, I'm currently the director of the University of Oregon's Charleston Marine Life Center, located here in Charleston. And we opened in 2016, so just a little over 4 years ago. Prior to, I'll show you a picture of what we look like here. Oops, there we go. I'm not able to see all your faces right now, so I was going to ask how many had been out here, but since I won't be able to get those answers, I hope many of you have had a chance to be out here, and if not, that when we reopen, you can again. We are currently closed for coronavirus considerations. So we hope that that won't last too much longer, but just so you all are aware, we are under not only the county and state regulations, but also the University of Oregon's regulations for evaluations of when we can safely reopen. So it's a big balancing act, but particularly because, as you will see as we get into the presentation, we are structured for a lot of interaction, specifically geared to encourage that hands-on exploration and interaction. It just makes it all that much more tricky to be in a position where we can open safely. So we're not there yet. And all I can say is to please follow our either social media or our website, because we'll be posting updates there. But when we were open, we were seeing about 15,000 a year in the center. And I'll just take you inside. So the goal for this presentation is to show you around, say what it looks like, introduce you to some of the animals that we have on display, some of the exhibits that we have, some of the activities that in a normal open stage that we offer the community. And then because you guys are funding a new exhibit for us, I want to show you where we are on that exhibit. And all throughout, thank you for that very much. But we're going to get to that after I show you a little bit of this. So a little bit of just orientation for the center. As I mentioned, we're part of the University of Oregon's Oregon Institute of Marine Biology, which, if you aren't familiar, U of O has had a marine field station here in Charleston for decades. It started in the 20s, actually, as a summer field camp with tents at Sunset Bay, and then has gone through various growth since then to multiple, both teaching and research buildings, housing, cafeteria, library, et cetera, out here in Charleston. The Marine Life Center itself is the new part of that. And as I said, we opened in 2016. We're kind of a blend between an aquarium and a museum. We have aspects of both. And we attract both local residents to the center and visitors and school groups throughout the whole southwest region of Oregon. Okay, so what you see here on this screen is some of the downstairs. We have various aquaria. We highlight local marine organisms. Our main purpose is to be an education resource, and we use local marine biodiversity to do that. We often get the question of, are these animals found here? Anything that we have in the tank was collected locally, so yes, those are here. So what's really fun is to get the exclamations we hear from people who have lived there their whole life and just didn't know that there was so much, right out there off the coast. Okay. All right, so I'm going to introduce you to a few of the animals themselves. Some fish here. The top left, it's a wolf eel. On the top right, we have rockfish. And then going clockwise there, that thing that looks like a rock with lips is a buffalo sculpin. And then the last one there, a kelp greenling. And you can see that it's actually laid eggs. So in our aquaria, we have them set up to kind of represent different habitat areas, and then it's really fun for those of us that are there a lot, as well as people that come in. It's fun because it's always different. And Kathy, I know, is on this I saw your name in here, and she could attest to that, that every time you come in here, something's different. Either somebody's laid eggs or somebody's grown up, or we've got new animals. So it's a good reason to keep coming back. Oops. Uh-oh. I don't know what happened there. All right. These little fish here on the left, you can see a pipefish. And if they remind you of seahorses from the face, it's because they are related to seahorses. They're essentially like a straightened seahorse. And they are local in our estuaries and in the bay. And like seahorses, some of you might be familiar that seahorses, it's the dads that give birth, in that they have the pouch, and the female deposits the eggs in the pouch in the male, and then they develop there. And then you can see, actually, and this was taken here at the Marine Life Center. We've had a few different pregnant dads, and you can see a little. Can you guys see my mouse? Mm-hmm. Okay. Yes. So you can see the little baby pipefish. Anemones, lots of different anemones. I'll use my mouse then. The green anemone here, this is a white spotted rose anemone, all closed up to look like a package. This one, I wanted to have a picture of this because this has happened a few times with us. And do you see here, and then another part of the body there? So this type of anemone, called the aggregating anemone, it can literally pull itself into two. And that's, they do that frequently. And if you've ever been out in the tide pools around here, and you see like a patch of identical looking anemones over here, and another patch right next to it, and there might be like a ½ inch of space between the two patches. What that is two separate clones, essentially. You had over here an anemone that split, and then they split, and then they split, and then they split, so they're identical. And a different one over here did that. So two separate clones, and then that little line is essentially a demilitarized zone, because on their tentacles here, they have little barbs, and they can sense another of the same clone, and they don't attack. But if it's a different clone, they will. So the neighbors on there, if this is making sense, it's hard for me because I can't, when my screen's up, I don't see any of your faces, so I'm not sure if this is making sense. But as one clone is fighting another clone, they back off just a little bit and leave this space in between. Okay, I will say, if you have questions, I can't see you, but if you unmute yourself and say something, please feel free to interrupt me at any time with any questions.
But Trish, I think we're all following you, and we will have questions afterwards. So.
Okay. But I don't mind if something's related to a picture, if somebody wants to ask.
Somebody got a question now?
Am I coming through?
Yes.
Yes.
No, this is Jim Layport. I was just going to say that I don't remember how many years ago, probably 30, my daughter, when she was out there at the Marine Station, did the research on those cloned anemones.
Fantastic.
And she has movies of what she did and how that they actually fight each other.
Yeah.
She has movies of the tentacles from one clone attacking the tentacles of another clone. And,
that's fantastic. Yeah, I would love to see some of those videos. Yeah.
Well, I think she probably still has them in her archives somewhere, but yeah, she did. She was actually doing a photography workshop at the time that she did that. That they did that research, but she was attending Stanford and did a summer program out here and did the stuff on that on those anemones, which.
Yeah, that's great. And
I didn't know anemones had war.
They do. Yeah, it's amazing. And that's a good point to bring out as well, that OIMB does have people that come from all over the world to do research here and stay in the facilities and use the facilities here. But so anyway, these anemones is just about to split there, as you can see. And being able to clone is something that I would like to be able to do some days, definitely. This anemone over here, common name proliferating anemone, what you see here are little baby anemones on the side. So an anemone is essentially a bag. with one opening. So anything that's going in or out of that animal, it's in the same hole. And that actually is what we tell people when we see them at the touch tank poking in the anemones a bit too much. We can remind them, or just ask them if they realize that that one opening is not just the mouth, it's also where anything comes out. And that's a good technique to get some kids to stop poking the anemones. But so whereas most anemones are what's called broadcast spawners, so they release gametes, sperm, and eggs, and they fertilize in the water column, turn into larvae, and then metamorphose into the adults. This is a brooder, so they actually hold the eggs inside the sac of the anemone, and then the little baby anemones, when they're ready, come right out and crawl and hang out for a while on mom down here before they crawl away. So kind of fun. Okay. Sea slugs. So no doubt some of you are familiar with these animals. They're gorgeous. I think certainly ever so much more gorgeous than the slugs I've been battling in my garden this spring. But sea slugs come in all sorts of colors, different frilly aspects on them. And if back to the point about war, these animals are amazing at warfare. Not all of them, but some of them will take those similar stinging cells out of things they eat. So they might eat other animals that have stinging cells, and they can somehow, instead of, when they eat them, instead of the stinging cell firing and harming them, they're able to ingest it and keep it from firing. They ingest that stinging cell from the other animal, and it goes through their digestion and then up through, and then maybe at the tips, it's different species, but at the tips of the cirri here, and then they have essentially stolen the weapons from another animal, and they protect them from being eaten. So pretty remarkable in that. These were all taken, all the pictures of the animals in the show, I will say, I took in the center. So these are all animals we've had at different times in the center. This here, you can see this little nudibranch is laying eggs. And then you can see further along with more eggs laid there. So I'll point out that this, it's not just in this picture, this nudibranch. This is a nudibranch. This thing that looks like a pink shag rug. Okay, stars and such, I'll say. So we have the typical sea star here. And then also in the same phylum, Echinoderm, there are brittle stars with long, slender arms. There are basket stars here, which looks like a brittle star, but the arms are highly branched and curled. Like with all of these, the mouth is on the underside here, but this animal moves those arms through the water, catching small particles out of the water, little hooks along the arms, and then moves the food down and around and down and finally to the mouth right there. This one, back to the top right, this is called a brevispinus, and it specializes on eating clams. I took this picture because it was right on the glass, and you could see that underside of the ventral side. But do you see how the tube feet, so these all move by tube feet, which is like a hydrostatic system that pumps water through. But the tube feet in the center area around its mouth are longer. And what it does is it crawls over the sediment, senses a clam, and then it can pump more water into those tube feet to extend the tube feet, to make them even longer. And then it reaches down into the hole where a clam is with those lengthened tube feet and pulls the clam up out of the hole. And then, oops, sorry, you can go back, can feed on the clam that way. I took this picture, this is a bat star, but when this, when a lot of stars, when they get what they're eating, the bivalve or the clam, the mussel, or like I said in that one, usually clams, it's use the tube feet to hold onto both sides of the shell, both valves. It's not strong enough to just pull it open, but it holds it in tension. And eventually that clam or mussel will tire a bit and the mussel will relax, and that tension that the star is putting on it can pull it open just a little bit more. And it does that in that battle of holding and pulling and holding and pulling until it can open the valves just enough that the star will then, the stomach of the star, it will eject its stomach out of its own mouth and through into the crack of that slightly open clam or mussel, digest that clam meat or mussel meat inside the clam or mussel itself, and then pull its stomach back in. So that's pretty remarkable. So this shows the stomach partially out. So that, and this was just a fun one showing how they can turn themselves over. But it looked like it was doing yoga. All right. We also do try as often as we can to have octopuses in there. 2 different species around here that we have on display at different times. The upper left one is the small Pacific red, and the larger one there is yet a small giant Pacific octopus. We can only keep them for a certain length of time because they can grow to 20, 24, 25 feet, obviously bigger than our tanks. This one was Hank, if any of you remember Hank. Okay, crabs, lots of different crabs. The lower right, can everybody see the crab here? There's a claw. This is called a decorator crab. So it has little barbs, kind of Velcro-like barbs on its carapace that it then goes around with its claws and grabs other living things and sticks them to itself, whether it's algae or little anemones or bryozoans, and it covers itself with whatever's in the area to hide itself. So the crab here, you can just kind of tell by this, but this whole thing is crab covered with other stuff. This is actually a movie that I'm going to play for you. So I just by chance caught a red rock crab molting. So when a crab molts, many of you are probably familiar with this, but just in case some aren't, to grow, since a crab is in a hard shell, to grow, they have to first themselves grow a new shell, and it's under the old one. So in order to be bigger, it's kind of soft and folded up a bit. And when they're ready to molt, they will take water in to their bodies, which expands the new soft shell to the point of cracking the old hard shell. And then they just crawl out of their old shell. So I took a time lapse of this process. Obviously, I didn't catch it at the very beginning, but let's play this. Oh, wait. Hold on. Try that again. There we go. So it's taking in water. There. Did we get to see that? Yep. Okay, so you could see how much bigger it was when it came out, but then its body is really soft. So it's very vulnerable to being attacked by other things, so they tend to then go hide for a while until the shell hardens. So oftentimes if you see crab shells on the beach, they're actually the molts of crabs. And also if you ever have eaten soft shell crab at a restaurant, what you're eating is a newly molted crab where the shell is still soft. Okay. Mole crabs, so related to them, let's see if this video works. I love these little guys. They have antennas and they are catching little food particles in the water, and then they swipe them past their mouths to eat. And these are found on sandy beaches, kind of right at the tide lines, and they kind of move in and out with the tide. And when they swim,
Oh, look at them.
There you go. Okay. All right, so that's just some of the animals that we have on display. I want to show a little bit of what we mean by an exhibit as well, since you guys are involved in our larval exhibit. So we try to have our exhibits have interactive components as well as different visual and a mix of, say, video and specimens. So when we highlight deep sea for our deep sea exhibit here, we have deep sea specimens that are preserved. This is the deep sea version of your backyard pill bug, right there. Little isopod, and this is a lancet fish, which is caught off the coast here. So this shows that area that focuses on deep sea. So you see we have the specimens, we have live animals highlighting here, some here. We have video. This is video that cycles through different areas off the coast, the reserves off the coast, but showing what's living on the benthic, the bottom area, which people can't see for the most part. I mean, if they even go out there on a boat, they're just looking at the surface. So this is, you know, taking you under to see what's there. And then this video here is one of my favorites. It's taken off the axial seamount off the coast of Oregon, which is a hydrothermal vent. And people, you know, if they're even familiar with the hydrothermal vents, often think of Galapagos, but we have them off Oregon as well. So that's where super hot, mineral-rich water, and when I say super hot, I mean like about 750 degrees Fahrenheit. It's coming out at the ocean bottom, like I said, mineral-rich, hits that cold, nearly freezing seawater around it. So all those minerals precipitate out and build chimneys. And this video, as it cycles through, shows those chimneys and shows that even in that area where you have this hot chemical soupy mix, there are animals living there. And so that video runs through, it shows you those animals, and then we, have animals in the tanks here and content around that. So that mix. Another example for fisheries. Obviously, fisheries is very important in the local community, and we want to highlight that in many different ways. So in this case, we utilized the windows of the center to provide a view and an access that most people can't see easily. There aren't a lot of places to get these views coming out to Charleston, but our windows overlook the Pacific Seafood Offloading Center, and they've just been fantastic to work with. So we can see the boats being unloaded. This is a squid boat, so you could look out the window and they have these giant pumps which go down into the hold to pull up the squid catch or the crab, not just when the crabs come in, but you can also see the equipment being loaded onto the boats. And down here, actually see right down into the hold of the boats to see that those are all pink shrimp, by the way. And then this, if you turn out to a different view of the window, you can see when they're unloading the fish and sorting and weighing and processing, and then also they have different regulators come through from, it's a mixture of ODFW and NOAA, and even state police have different roles of checking there. So it's a highly regulated, monitored fisheries that our visitors can see, you know, taking place. We have a whole open air room where we have other tanks with different animals from crabs to lingcod and rockfish. We have a display where Chief Brainerd made some replicas of Native American fishing tools for us, which is really incredibly beautiful to see. And then we have touchscreens where people can get detailed information of different local fisheries and fishing techniques, time of year that it's done, and all of that. So that's for fisheries. Our newest exhibit actually is on birds, on marine and shore birds. So again, this is a case where we've utilized a window, have spotting scopes, a guide of what's normally seen right in the bay, and then also a journal so people can record what they're seeing. We have cases with the birds here, and then these drawers that people can open and get a lot more content. So like this drawer, for instance, has skulls of lots of different bird heads, but what's striking is to see all the different beaks and the patterns of the beaks, and then learn how that allows them to get different food. So This is a good example to point out. Our goal is when we have these exhibits is to make them accessible at all levels. So obviously someone who's going to come in and spend a lot of time reading and looking at the details of the beaks, that's one thing. But then another drawer has bird bones that you can lift up and feel how light they are, or look at different feathers. So again, you know, you could have a young child at this drawer, well, you know, the adult is over here at this drawer. So we're trying to provide that variety. And you can see here the spotting scopes are clearly popular with all ages. Okay. I think this is the last one to show just exhibit example for marine mammals. A lot of the upstairs devoted to marine mammals from we have complete skeletons of both an orca and a young gray whale. We have big pieces of baleen that's in here. That's what's in the, you know, some whales have teeth, like this orca here. This gray whale would not have teeth. It would have baleen that allows it to filter out food. So there's a lot on that. But then you can see other skeletons back here of different local smaller whales, or called porpoises or other things. Also seals and sea lions, the evolution of marine mammals. Here's the skull of a sperm whale. Some of you might remember in the late 70s when there was a mass stranding of sperm whales up off Florence. So about 40 whales stranded at that time. This skull is from that, but you can actually go and touch that, knock on it, see what parts are hollow or not, look for where the brain would be, which is not where you think it would be. Then this kiosk here is super popular. It actually allows people to touch a whale and hear the sounds of that whale, but not just hear it, they can then record themselves trying to imitate that sound, and then it plays a spectrograph or a picture of sound back to show how well they were able to match that sound. And then there's also a lot of information in that kiosk about research on the different whales, conservation status, different aspects. So a lot in one station. Okay. Last two slides before we talk about larvae and your exhibit. Just wanted to point out different ways that kids and school groups utilize the space. This room here, you'll see it's folding chairs, folding tables. That's intentional. So we can set it up in different ways for different programs and different activities and bring out material for different grade level classes. This is an example of a class where they learn a lot about the skeletons, but also what you can tell from looking at a skeleton. So trying to help them understand by looking at whether a bone has extensions on it, what that means. That means that's where muscle would attach. So then being able to see this, you can kind of get an idea of how something can move. You can also look for clues of how the animal lived by other aspects of the bones as well. So we have them do that, but we have them look at the skeletons we have on display, and then we bring out a tub of bones and they have to put it together and figure out what it is. We also have integration of art as well. So sometimes schools come in with different programs and are doing a lot of sketching as well. When we can, we bring in live videos from research trips. So this is a feed of a deep sea research cruise going on there in that screen. It's not just school groups. We have different programs for the public. This center one down here, you see adults in the chairs here, but then there are all these younger kids here. We collaborate with the Coos Bay Library and do a story time out here. We've been doing that about twice a year. So they'll read marine themed stories, and then we'll bring out the specimens for the kids to see what they've been reading about, and then they get a tour of the center as well to see the animals in the center. This is a photography group that came through. So they were learning skills of photography in aquaria and what that takes, as well as with moving animals. Here we have different local art groups come in and sign up for a time in the center to paint and draw. This is an adult education group that came in, and they did the same program that we have for the kids with the the skeletons as well, but we offer those for adult groups as well. Then these last two pictures, each year we've been having a free night at the aquarium Halloween party for families. And so these are pictures from that. So it's kind of mixing a lot of fun activities and crafts with, you can see the decor all around. We decorate the center and have dry ice making steam come out of the touch tank, and we have carved pumpkins in the tank so that fish and octopus can go in and out of the pumpkins and things like that. So it's a lot of fun. Okay, so exhibits in the works. The larval exhibit that I've mentioned, that we're super grateful for your support on that. We also are installing a jelly tank, for a jellyfish tank. And then we are also installing a touch screen where we can have content of local research. So for instance, the graduate students or research faculty across the street can develop short videos that will highlight work going on there. And those are things that while we're closed, we're still busy over here because we're working on all those things. In the future, when we get more funding, these are two exhibits that have been identified that we want to have in the future. One is one on the impacts of marine biology research, because a lot of people will think marine biology and they might think, oh, dolphins are pretty or fun to watch and other stuff is pretty. But what a lot of people don't realize that I think is important to get out there is everything that's come out of the study of marine animals and the ways that touches our everyday lives, whether it's through medicine. There's a lot of research currently going on cancer fighting drugs in sponges. Cone shells have a toxin that has a lot of interest going into it now as a substitute for opioids. So it's a pain reliever that is not addictive. So there's that medical, there's a lot on for neuroscience and development. Squid have like the largest axon from their eye, so has been a tool for a lot of studies for vision. Sea urchins have been key in understanding cell biology development. So there are a lot of different examples, and that just is just touching on it, but even into engineering. I think what's interesting to think about that the design of skyscrapers was partly built on analyzing and looking at the structure of spicules in glass sponges. So there are so many connections that just aren't well known of how the study of marine science affects our lives. So I'd love to have an exhibit on that. And then kind of one of the habitats around here that's so common around here and yet we don't have an exhibit on are sandy beaches and rocky shores. So, okay, but let's talk about larvae. This is my biggest word slide, so I'll give you a chance to look at that. But kind of the main points of what we're hoping to do with this exhibit is use larvae as a hook. Larvae are the babies of many marine animals. So people are familiar with tadpoles becoming frogs or caterpillars becoming butterflies. And in that sense, you have a baby that doesn't look like the adult, and there's a metamorphosis to the adult stage. The marine invertebrates are like the big example of that strategy of development, that from one stage a metamorphosis to another stage. So there's an estimated 170,000 marine invertebrates that go through this larval to metamorphosis to adult stage. And so even a lot of the animals we're familiar with, like crabs and sea stars and snails, their babies don't look like them. They don't live where the adults live, and they float around in the water for a long time, so they're carried different places. So there's this whole break between the baby stage and the adult stage that has a lot of implications. So, but the other thing here, when I say it's an engaging hook, larvae come in so many bizarre shapes that they're a good way to engage people in learning about various aspects of marine biology. because when you study the larvae have connections to embryology or development, genetics, who eats who, how things are managed. Like I mentioned, if your babies are in one area and the adults are in another area, if you're looking at any kind of fisheries management, you need that whole picture. So there's lots of reasons to know about larvae. But the shapes, if some of them look familiar to people, they have actually been the inspiration for a lot of science fiction movies for what aliens or spaceships or whatever look like, whether in Star Wars or James Cameron is well known for using marine larvae for a lot of his design ideas. So, and the other thing is to let people know that right here locally, OIMB is a world-renowned center of larval research, and that isn't known by a lot of people, so it'd be nice to get that message out. Having our center and adding to it and bringing in ways to engage in different topics, but to different age groups, different programs, all of that is building a resource for the area, whether it's to support the schools, homeschool as well, continued ongoing learning. That is one, but it also, and this is something I pointed out in the proposal, is I feel strongly that we can contribute just as a piece, but a thriving community needs, the museums and the science centers and concerts and performances and lectures and restaurants and all of those things that we're all missing right now as we're staying home. But all those things contribute to the thriving community and help in efforts to recruit businesses, families to the area. So while this is just a tiny piece of that, it is a piece of that. So thank you for your support again. So let's see what this larval exhibit looks like. This is the space itself as it looks currently. So it's mostly empty, as you can see. We have 2 screens in there, and this one has beautiful footage. It's stunning high definition video, so through microscopes by George Fondassa, one of our faculty members, of these teeny tiny larvae, because if I didn't say it earlier, most of them are not visible to the naked eye, or if they are, it's just the teeniest speck and you can't really see what it is. So this is letting people see what they look like. And then we just repeat some of that video over here for now. But we have all this hope for this center, for the rest of that space. So let me show you where we are, because it's coming along really nicely. This is a SketchUp of what we are working on. So this would be the left wall. Let me go back. So left wall, center wall, and right wall, okay? So the left wall, the center, and the right. So you don't have to look at all the details necessarily, but it's like in the other exhibit, it's a mix. We want both visual, tactile, some video here. This will stay, that video that I talked about. This video over here is going to change, and I'm going to show you what we've created for that, which this one is really for our younger, but I think all ages are going to enjoy the challenge, but something accessible to the younger, down low. It's Who's My Mom? And we'll look at that in just a minute, the video itself. Drawers here that will have models of larvae, and I'm hoping you can still see me, but so we're doing 3D prints of larvae so that you can actually feel them, pick them up. Again, remember they're microscopic specks, so you wouldn't see it all, but see they come in many different shapes. So those are the drawers, along with some props on the physics, because when something is that tiny, the whole world is different. It's like inertia goes away, and the seawater that we can move our arm through easily is so viscous at that scale that it would be like pushing through something thick. So imagine if you have a particle of food here and you've got a cilia and you're trying to catch that food, it would essentially just move that package of water, thick water would just push it away. So there are all these designs to be able to capture food and swim in this thick viscous fluid. So we want people to be able to experience that some as well. The screens we'll talk about first is what is a larva, you know, why is it important to know about larvae? Some of the things I've been talking about. How do you study larvae and how that's changed over time? So there'll be a history component too, because until you had microscopes and the right kind of nets to catch these things, it was a mystery, you know, how these other animals reproduced, where were their babies, things like that. So go through some of that history too. So that allows people, some visitors be interested in the biology, some might be interested in the technology side of it, some, as I mentioned, on the physics aspects of it. So it'll be a little bit of everything in that regard. A plankton net, what we're thinking now is actually changing this a little bit to have an area where different materials hang down, because different things were tried at different times as ways to catch plankton. Have a stand here with maybe some old microscopes, the early types, life cycle diagrams here. Anyway, you get the point. It will be a mixture of ways of presenting the content. And at this point, we have most of the text flushed out, a lot of the photos taken. We've identified others that we either still need or illustrations to present that. Like I said, we're making these models, getting the nets. We've made this video here, the challenge video, so we can see some of that. Those are those models again. The video here. So this will be as a video sense, but in this presentation, you're going to see different slides. But you saw the larva and the challenge of who it was, and then you'll be able to see what a baby worm looks like. It doesn't look like a worm. So that does not look like a worm, but that is the baby worm. So again, up top, whose baby is this? Or what is the adult down here? Is it an anemone, a sea cucumber, or a bat star? That's what a baby bat star looks like. Any guesses? Yeah. And this other bonus information here, this is our state shell, the Oregon Triton. So is that a baby octopus or a baby squid or a baby rockfish? Okay. So that's a flavor of the exhibit and the different parts. It's coming along really well. I do want to just put my contact information up there as well. Please don't hesitate to reach out. I would not use that phone number right now though. I'm not often at my desk. I come in, check the animals, work on the water and things like that, but I'm mostly working from home right now, so shoot me an email instead.
Thank you, Trish. That was amazing. It's fascinating. That kept coming up in the chat room that it's fascinating.
Okay, I'm gonna close the presentation so I can see people's faces again.
Okay. So questions, anybody?
I failed the larva test. Flunked.
You flunked the larval test?
Flunked.
Wow. Sorry, I'm just getting the screen up to see your faces.
So anybody else with a question or anything to say? Jim, you
I
have
have a
something?
comment. Oh, I'm sorry. Go ahead.
He's not unmuted. Unmute yourself. Okay, there you go, Jim.
Am I unmuted?
Yes.
Okay, you were mentioning water there. Where do you get your clean seawater from for the station there? Because I would think that the bay would be variable to saltwater content, sediment, pollution and whatnot. Do you take water from the ocean somewhere?
Yeah, no, that's an excellent question. So we pump water near the mouth of the bay, and we do that intentionally there and timed to incoming tides, because we don't want bay water going out. We want that ocean water coming in. So it's pumped twice a day, timed to the incoming tides, and that water gets pumped holding tanks up the hill. And those tanks are just, refilled twice a day, but then it's a constant gravity feed. So it's a running seawater system. We're constantly getting fresh water going through the tanks. And it's the same system that all of OIMB is on. So in their research labs and teaching labs, their tanks are getting that as well. And then it's a balance that we play with the water at that point. Mostly we then use unfiltered water in our exhibits. And we do that because that water, those larvae that I mentioned, but also then phytoplankton, other plankton, they are, one of the things they are, food for other animals. So if we want to be able to keep a lot of the animals that filter feed or by eating that small food out of the water in our exhibits, we need that plankton coming through to feed them. On the other hand, when we have a big storm and things get stirred up, even though we're pumping near the mouth and incoming and all the rest of it, it gets stirred up, and sometimes we get these sediment pulses. So we have a sign by our tanks that says, basically saying, we do take care of our tanks, but sometimes when you see it cloudier, that's natural, that's normal, and it's healthier than if we had a strong filter and filtered everything out of the water and it looked crystal clear, but then we'd have to supplement all the food stuff. So.
Another question from Mike Gaudette.
I was just going to ask if she got good interest from the public schools, the elementary and high schools in the area.
Yes, yes, we do. Both local schools, and that I expected. What surprised me a little bit, but then when I thought about it later, it shouldn't have surprised me, is we've had significant interest in schools from Roseburg, Grants Pass, Ashland, a little further than I would have thought. But as they explained, we are a challenge for a one-day field trip for them, but we are doable. We're going just that more, that further to Newport, for instance, just makes it impossible, or they'd have to do an overnight. So not only are we serving Coos Bay, North End, Bandon, Coquille, Myrtle Point, all the ones you would expect, but we are actually pulling a number of schools from a little bit further as well.
So Mike Gaudette, you have a question?
My question was, do you track your visitors to see where they're coming from? And how many are local from Oregon or versus out of state and international?
Yeah, we don't have exact numbers. We have a log book at the check-in desk, and we encourage people to sign in. And one of the questions, if they do it, is where they're from. So not everybody does that, but we get a good number. So I haven't gone through and done a count per se, but I have frequently flipped through to see where people are coming from. And obviously we get people locally here. We get a lot throughout the state, but we have had people from, I should go back and check if it is every state, but it's certainly almost every state, if not all of them, have come through, and several international as well. Those increase in the summer, not surprisingly, in terms of when the people are traveling the Oregon coast and stopping through. But there is a large number of people coming out of Oregon.
Eva?
Tom, yes, go ahead.
I sure have enjoyed Trish's presentation, and wish that we could continue with questions and answers. But our brand new member that needs to be inducted today has an appointment coming up at his office. And so.
Okay, so Trish, thank you very much. You're welcome to stay and hear this new member being inducted. But
Okay.
this was a great
I'll do that.
presentation.
And for any of you I didn't get to answer questions, like I said, you have my email, so please reach out to me.
Right, thank you so much.
Thank you.
So Tom and Patty, you want to go ahead with our first induction in the COVID times?
Yeah, we're Rotary, as ever, the creative organization. We have figured out how to bring in a new member, and we want to induct today. Mark James, who is sponsored by Tom Bennett, and three of us are here in his office watching the presentation. So President Eva, fellow Rotarians, and any guests on today, we want to welcome into our club and admit for membership Mark James. And Mark he's a realtor in town and has a history of being in the area. Had a chance to spend some time chatting with him, as has a number of our folks from membership and board members. So Mark, you have been elected to membership in this club because you possess qualities that will help you to portray the spirit of Rotary in your public, personal, social, and private lives. We encourage you to attend meetings, share in club service projects, participate in social functions when we get to have them again, and volunteer your time, knowledge, and talents to the club as you carry out the principles of Rotary into your daily life and into the community. Your membership in this club entitles you to be welcome as a guest at any Rotary club in the world and gives you the opportunity to volunteer for national, international projects, and hopefully a lot of volunteering locally with our club. So in the name of the Rotary Club of Coos Bay, North Bend, Oregon, we now formally admit you to membership of this club. And President Eva and fellow Rotarians and guests, please help me welcome our newest member, Mark James, into the club.
Yay.
And he's getting his pin and badge. Woohoo!
I'm keeping the badge because I've got the box.
Haven't seen a badge in a while.
Someone must have them locked away somewhere.
Yeah.
Welcome to the club, Mark.
Well, thank you. It's an
Congratulations.
honor and a privilege to join you. I wish my wife could have been here, but she's probably delivering a baby at this moment. So that was the plan. And she's a physician at North Bend Medical Center. A local gal, born and raised here. And we have kind of transformed our family. I have 3 kids. I met her in college in Portland, a Navy life, and I'm sure I can get more involved in, we'll get to know each other a little bit more, but I have 3 kids. 2 of them have graduated from college. One's a junior in high school here, and I'm busy. I'm busy here, and I look forward to having this organization help me reach out to other folks and to yourself and especially in the community. So again, it's an honor and a privilege to join, and I appreciate your time.
We're glad to hear your autobiography too. That's great. Got it all in one shot. It's wonderful. Anybody have any questions for Mark? Where were you born? You said your wife was born here.
Well, I'm an Air Force brat. I was born in Colorado, but I grew up in East County. We call it East County, so that's Gresham, Portland, Southeast Portland, and so that's where I grew up. Went to college, played baseball. My wife was a volleyball player from Marshfield back in the day, and then we met in an athletic training class. There you go.
Hey, great. Hey. Any other questions for Mark? Okay, welcome to you. You're the very first new member in the COVID time. Amazing. You've made your mark. Mark, you made your mark.
Well, thank you.
Anybody else? Okay, well, Tim Navani, Bill Grau, Deanne, and I don't know if anybody else has, but we've been attending the Rotary Virtual Convention. Oh, Angie, you have too. Great. Okay. So yeah, we've heard some very interesting programs over the last few days. It continues until Friday. Tim, what have you heard? What have you been listening to?
Well, just a brief summation. The general sessions were very interesting, fantastic given the fact that they were done for the first time ever virtually. We heard from, of course, Rotary International President Mark Maloney, and had a big salute on day 1 to Rotary's connection with the United Nations. Of course, the big anniversary coming up on Friday or Thursday or Friday, June 25th, 1945 is the anniversary of the signing of the UN Charter, which Rotarians played a big role in. If you don't know, you can find out more online. But Secretary General of the United Nations, Antonio Guterres, actually made a brief presentation online to the Rotarians at the convention. Foundation Trustee Chair Gary Huang, who was actually president, Rotary International president the last time I was president, gave a talk. And we heard some great Australian pop opera from Arias. Holger Knack talked on day 2 of the general session, and we talked about Rotary clubs leveraging new platforms to meet virtually and stay connected, which is what we're doing. We also heard from Rotary Peace Fellows, lots of stuff going on in the sessions. I know Bill has talked about some great stuff that he's heard in some of the general sessions. Bill, do you have anything you want to pass along on that?
Well, on one of the breakout sessions yesterday morning at 6 in the morning, mind you, there was a really interesting discussion about using social media and other techniques to stay engaged during this COVID era. And one of them was a guy that's the editor of Rotary in Great Britain and Ireland had a session on, or a segment on whether it was prudent to live stream club meetings as a way to do hybrid get-togethers. And I'm kind of persuaded by his logic that the way to do hybrid meetings is not to live stream and have somebody see a video of us munching down a sandwich, but rather to have a mixture of some live meetings in person and some hybrid meetings where we meet through Zoom. But that's for the board and the club to ponder over and think about going forward.
We also heard about reducing our carbon footprint, which was pretty cool, with all the different ways that we can do that. And our bags, our sling bags, are right up that alley.
They've got that walking
challenge
walking challenge going on. Have you looked at how many steps some of these people have taken? Some guy or gal, one day into this, had 70,000 steps. And I thought, what? That's impossible.
I dropped that.
That might be a 4-way test issue.
I dropped out of that challenge. I started it and said, I can't do that. Not possible.
Yeah, I would probably never get out of my chair again, because I would just say, like, I've got no chance. So I just.
And you can see that Tim and I are in the same place. We're both in Hawaii here. See our background? Yeah.
We're in virtual Hawaii.
We made it.
But it goes on through Friday, and there are also going to be some breakout sessions all through the month of July. And so it's going to be a lot of stuff that is still pouring out, a lot of great information. And so if you get a chance, you can check those out, and we'll, of course, bring some stuff to you as well.
All the sessions are taped and will be on Rotary International for anybody to see, every one of them, which is really cool too. So they're also going to do on Friday our relationship with Toastmasters in one of the breakout sessions, which is pretty cool because both Deanne and I are members of Toastmasters. So.
And before we lose anybody else, I want to take the moment too to have everybody give a big Rotary virtual cheer to soon to be past president Eva Shima-Takahara, who has put in yeoman's effort as president in what has been an unbelievable and tumultuous year with an unforeseen adversary in COVID, and has come out just in true Rotarian fashion. So Eva, you've done a great job paving the way for me going into my year, and I appreciate everything you've done leading the club and getting it ready for me, and I think you've done a fantastic job, and enjoy your soon reign as past president.
Thank you, Tim, and good luck to you for sure. Yeah, it's great. I mean, it's great that I'm leaving and you're taking over. That's what's great.
I know that feeling. I remember it.
Oh yeah. Oh boy. Okay. So next week, I want to hear from whoever is on about their year in this past year of Rotary. And if you've run a committee or whatever you've done or what you liked about Rotary in the past year or what you hope for the future. I've asked Jim Layport to talk about his time as a member because Jim is our longest serving member. Isn't that correct, Jim? Can you unmute?
Yes, yeah, I joined in 1966, so I'm 54 years
Wow.
at this point, yeah.
He's our longest serving member, so he'll tell us about his time in Rotary next week.
I'll be anxious to take some notes, Jim, if you got any good for incoming presidents.
I don't. I ducked the presidency 3 times, so.
Well, that would have been good information to have.
Before you got, yeah, I ducked presidency. But anyway.
Okay, and Tim, you'll introduce
the duck program chairman. I did that for a number of times.
So Tim, you can introduce the new board, which has just one member leaving, which is Stephanie, and Al Solomon coming in, right? Is that correct? I think so.
Yeah, Al's coming in. Yep.
And Stephanie is retiring. So
yes. Well,
anything else put of the order from anybody? Nope. Okay. Thank you all. See you next week. Okay. Bye-bye.
Mahalo.
Mahalo.